Hello again. So we talked about naturally occurring clones. We talked about um, monozygotic twins, which I have here, but it's not. Don't worry about that yet. So we talked about monozygotic twins. We talked about organisms who lose an arm and grow a body. We talked about plants. We talked about those organisms that reproduce asexually. So let's talk about two artificial ways. So one artificial way of doing this is basically creating monozygotic twins. So what you can do is a long time ago, you were one cell big, and then you had a division, and then you were two cells big. You probably don't remember. And then you had another division, so now you're four cell big, four cells big. What we can do is we can break these cells up. So here you actually see a developing embryo, and they're starting to suck the cells out. And you can just move them to different locations. And now that they're no longer joined, they will all become individuals. So that's one way of creating clones, by breaking up the cells in this developing embryo. That's a little bit simpler, maybe a little bit barbaric. Hopefully they're not doing this with humans. It would be hard because you would need to put those back into uh, a mother to carry it to term for birth. Um, but a bit more complicated version of making clones is something called somatic cell nuclear transfer. And it sounds horribly confusing, but it's not. So we can use abbreviation, somatic cell nuclear transfer. And what this is that we're taking a somatic cell. So if you remember, there were two types of cells. We had gametes, which were sperm and egg, right? And then we had somatic cells, which were all the other ones. Those were like the cells that your cell in your cheek and the cell that makes up your muscles. And we're going to transfer the nucleus into another cell. So let's demonstrate this, and let's pretend that we're cloning you. A little strange, but let's just do that. So we have two cells here. The top one is, let's say, your cheek cell, and the bottom one is an unfertilized egg. So this is an egg down here, and this is your somatic cell because we got it from a cheek cell. The first thing we do is we enucleate this egg. So we remove this nucleus out of here. So now we have an egg without a nucleus. And then we transfer this nucleus into this egg. That's how you get the somatic cell nuclear transfer. So now we have an egg with your nucleus in it. And remember, your nucleus has all the information to make you, all 46 chromosomes, right? So then the next step is we need to transfer this new egg with your genetic material in there inside a willing woman. I'm not an artist, although you know this. And she will eventually give birth to you. So we will put this egg inside the uterus here. 9 months later, you are born. So there's like 3 individuals involved here for a somatic cell nuclear transfer. I heard they did this with humans. I hope it's not true, but there was some rumor that they did. So the 3 different individuals is you, right? You have to donate your somatic cell with that nucleus. The other individual has to donate the egg, which will be removed. And the third individual needs to give birth to you, needs to carry you to term. So that when you look at this example for Dolly the sheep, there was the cell that was donated. There was the one where the egg was enucleated. This was eventually put inside a third sheep. And this sheep right here was related to, the clone of that sheep here. Unfortunately, Dolly didn't live very long because the genetic material was already the age of this sheep. So it died at a young age.